Dear students, this is Dr. Pooja Thakkar from Inside Biology. We are in the 9th session of human reproduction. In this particular session, we are discussing regarding oogenesis, that is the formation of mature female gamete in the ovary. The whole process is done in the ovary by 3 phases. First of all, we'll understand the diagram. On the left side, you will be able to notice the flow chart of oogenesis, into which you will be able to see the oogonium cell, which is basically diploid in nature. It can be also considered as germinal epithelial cells of the outer covering of the fetal ovary, which undergo mitosis, as you can see in the diagram, and that will form the primary oocyte that will get arrested in the prophase 1 phase. Before birth, this particular process is happening inside the primordial follicle, which are the primitive stage, or you can say a very delicate kind of follicles, which are holding oogonium or primary oocyte inside. Now, during the childhood period, the ovary remain inactive. You have to notice one point here, as no more oogonia are added after the birth. I mean, whatever number of primordial follicles are there in the particular fetal ovary, that remains as it is with the primary oocyte inside. Now, after birth, childhood during the ovary will remain inactive and it will get start maturing from the puberty period till the menopause comes. Okay, so now we can understand in growth phase is the ovary will attain maturity. The growth phase is proceeding, in which the female will reaches, where the female typically reaches the puberty, and that can extend for many years. Primary oocyte will then undergo cyclic changes. They'll do enlargement and maturation by obtaining the food from the surrounding growing follicles. The follicular cells are providing nourishment to the developing oocyte inside. You can see in the diagram that the primary oocyte will then follow meiosis 1 at the end of each 28-day cycle. And at the end of meiosis 1, it is producing one secondary oocyte and a very small first polar body. Polar body formation is super important because whenever the secondary oocyte will get any kind of disturbances in its cytoplasmic content, this particular first polar body will get fused with the secondary oocyte to provide nourishment to that. And the second most important point here is that if the primary oocyte is not at all undergoing meiosis process by forming smaller polar body and secondary oocyte, it will be a very big trouble, because at the end of meiosis only it will able to produce 23 numbers of chromosomes in each of the cells. So we need to maintain the haploid condition of the secondary oocyte here, and so for that reason we need to undergo meiosis 1 of primary oocyte to produce secondary oocyte, which is comparatively very bulky in size. And at this particular stage the process get a slight halt till the sperm enters into the fallopian tube. Okay, so in the maturation phase there are basically two kinds of events. The first and the most important thing is, if the secondary oocyte will notice the entry of sperm in the fallopian tube, then only it will finish the secondary meiosis or meiosis 2, and after that it will get fertilized with the sperm to form zygote, which is diploid in nature, because the sperm has the haploid content and the secondary oocyte is also having the haploid content. So the merging of cell occurs or nucleus occurs, and that will lead to formation of zygote, which is 2n in nature. At that time also, at the end of meiosis 2, the second polar body formation will take place. Then later both of the polar bodies, if not required now, they'll soon dies. So this is what happens during the case of fertilization. One important point we need to notice here is when meiosis 1 has been completed, you can see the follicular developmental stage simultaneously, that after puberty, at each and every month, each follicle is getting developed. That is then considered as primary follicle, which can form secondary follicle. After that, it can be considered as the later stages, or they can be considered as growing follicles. These follicles are basically present in the cortex region of the ovary. Later, when the secondary oocyte forms at the end of meiosis 1, at that particular time, that oocyte is perfectly ready for the fertilization process. I mean, that is perfectly nourished because of the presence of the surrounding follicular cells. You can see the diagram that the particular follicle which surrounds the secondary oocyte is considered as mature follicle, which is completely developed. The follicle is having follicular cells which are giving nourishment to the particular oocyte. Simultaneously, it is having the cavity which is showing in light blue color, that is filled with fluid, a particular kind of fluid which is formed by the cytoplasmic divisions of follicular cells, and the cavity is then known as antrum, which is fluid rich, that is also providing nourishment to the secondary oocyte. And after that, the ovulation process begin to release the ovum, or you can say the secondary oocyte, out of the follicle. For that, this particular mature follicle get attached to the ovarian wall, and then the secondary oocyte will be released from the ovary, which has been then captured by the fimbriary portion of the fallopian tube towards the infundibulum, and then towards the wider region which is ampulla. Now, the second most important thing, if the fertilization has not occurred, I mean, the secondary oocyte has to stay in its original position without the entry of sperm in the metaphase 2 condition, as you can see in the diagram, that will hold itself in metaphase 2 of meiosis 2. And then if the sperm doesn't come, or if sperms are not coming, this particular secondary oocyte will not be able to form zygote, right? So after that, it will go out as an unfertilized ovum through the vagina with the menstrual flow in the next menstrual cycle, or in the next menses. Then this particular structure which has been already ruptured, that is the follicular structure, which will be able to form a structure called corpus luteum. Now, after the formation of corpus luteum, there will be two positions again. There will be two conditions again, I'm sorry. The first condition is if the fertilization has been occurred previously, if zygote formation occurs, the corpus luteum will remain as it is to produce progesterone, because that particular progesterone will be super needed to maintain the pregnancy. Typically, we can say it will grow more and more blood vessels on the endometrium of the uterus to hold the fetus, or to implant the embryo in a particular site, as well as for the nourishmental purpose. But in case if the fertilization has not occurred, this particular corpus luteum will get degraded, that will get ruptured, and that will form corpus albicans, which is not at all releasing any kind of progesterone because no pregnancy is there. And finally, the process ends. So that in each of the condition of oogenesis, after childhood, you can say from puberty to menopause, every 28 days cycle begins, into which the first 4 days can be considered as menses. After that, the coming 14 days will be considered as the follicular phase. As you can see here, the follicles will grow and produce the mature secondary oocyte, and then the last phase will be considered as secretory phase, or it can also be considered as luteum phase, in which the corpus luteum formation occur. And after that, the two conditions will be there, as we have already discussed: the progesterone secretion in the case of pregnancy, and progesterone inhibition by forming corpus albicans. So it is a complicated situation, or you can say a process, which has been occurring in a particular female till the menopause will attain. Now, the next most important thing we need to understand here is the structure of ovum. A mature ovum is basically spherical in shape. The cytoplasmic content is super high. It can be considered as ooplasm, which is having a very large nucleus in it. The plasma membrane basically surrounds by the additional layer called zona pellucida that has been labeled here, and that zona pellucida, on the outer side, it is covered with the kinds of cells called corona radiata cells. These kinds of cells are having multiple projection on its surface, which are having basically tapering end for the attachment of the particular ovum to the fallopian tube, as well as wherever it is moving in the particular female tract. The perivitelline space is present. You can see the vitelline membranes label. So the perivitelline space is basically present between zona pellucida and the plasma membrane, which is present inside. So this can be considered as the basic structure of ovum. Now, which kinds of hormones are present during the oogenesis process, which we have discussed earlier, right? So GnRH from the hypothalamus is stimulated, and that will stimulate gonadotrophins from the anterior pituitary gland. Now, every time this anterior pituitary gland is producing some kind of hormones to the particular organs. For example, here in this particular situation, we can say this particular gonadotrophin, which are released from the anterior pituitary gland, will transfer towards the gonads to produce FSH and LH. So FSH, that is follicular stimulating hormone in female, will stimulate the ovarian follicle development and division of the primary oocyte towards the secondary oocyte. I mean, that is helping in meiosis process. And the next thing is LH, that is luteinizing hormone, which is stimulating the corpus luteum to secrete progesterone, that in turn inhibit the GnRH by feedback inhibition.